Hello students. So today we'll be dealing with Unit 5 of your cell biology class. So in this class we'll be learning about cell culture. Cell culture is the process of growing cells under controlled conditions outside their normal environment. The cells are maintained or grown in a sterile culture vessel in a medium which supplements essential nutrients like amino acids, carbohydrates, vitamins, and minerals. They also provide growth factors, hormones, and gases such as oxygen and carbon dioxide, which are necessary for the cell's growth. The cell culture medium is also responsible for maintaining physiochemical environment such as pH, osmotic pressure, and temperature. So there are some of the terms that I need to introduce you to. In vivo means experiments conducted in the living animals or organisms. In vitro are experiments which are conducted outside the living system. In silico are experiments which are conducted in a computer simulation. Sterile means absence of contaminating microorganisms or other chemicals. So there are some basic essential equipments that are needed to perform cell culture. You will need a laminar airflow, which provides a sterile flow of air and it provides a sterile environment to maintain the culture. You'll need culture medium for supplementing minerals, nutrients, and gases to the cell. You will need a cell culture vessel, which is a sterile containment for the cells to attach and grow on. We will also need a microscope to observe the cells under magnified conditions. There are two types of cell culture: primary cells and cell lines. So primary cells are isolated directly from human or animal tissue. Primary cells can be widely classified into adherent or suspension cells. Adherent cells require an anchorage and are termed as attachment-dependent cells. Suspension cells are attachment-independent cells. An example for adherent cells are fibroblasts, adipocytes, etc. Most of the cells that were isolated from the blood, such as monocytes or suspension cells. The primary cell culture is initiated from tissues using either an explant technique, where the tissues are mechanically dissociated and made to release the cells, or the tissues can be enzymatically digested using enzymes such as trypsin or collagenase, which again releases the cells. These primary cell culture may not be homogeneous because it may contain all the cells that are present in the tissue, and they possess very limited replicative capacity. So the cells that have been cultured for a long period of time are called cell lines. They have achieved, due to long period of culturing, have achieved homogeneous genotypic and phenotypic characteristics. These cells can replicate indefinitely. Though some cells acquire this property by themselves, cell lines are usually genetically modified for longer replication. Cells isolated from cancer tissues and stem cells can replicate indefinitely by their own. Other cell lines are induced with cancerous or stem cell genes. These cells are known as immortalized cell lines. For example, iPSCs, induced pluripotent stem cells, are transfected with pluripotency associated genes which are present in the embryonic stem cells such as OCT4, SOX2, NANOG, and CMYC. It is imperative that we characterize the cell lines. There are multiple methods of characterization. The first and the foremost method of characterization is observing their morphology. So observation of morphology is the simplest and the most direct technique to characterize the cell lines. Most cells in a culture can be divided into 5 basic categories based on their morphology. They can be fibroblastic, they can be epithelial-like, they can be lymphoblast-like, endothelial-like, or neuronal in their morphology. The fibroblast-like cells are bipolar or multipolar and have elongated shapes. They are attached cell lines. The epithelial-like cells are polygonal in shape and are attached, and they grow in discrete patches. The lymphoblast-like cells are spherical in shape and grow in suspension culture without any attachment. The endothelial-like cells are very flat and have a large central nucleus. They are also adherent or attached cell lines. Neuron-like cell lines, they have a very long axon and they are also attached cell lines. Thank you. So that will be the end of part 1 of your unit 5.